Also, I recognize I really have not made the time to put in a lot of poll questions, actually any poll questions this week. And I really enjoy those, and I've gotten always good feedback about those. We don't have any for today, but we do have some practice problems that I think will be beneficial. So protein and amino acid metabolism. So we're kind of on the last, well, we are. I'm going to jump over to, where did I put it? I think I threw a picture right up in here. We're on the last little bit of this whole figure. So if you guys remember this figure, we will have covered all the parts of this figure after today. So the main portion of this figure is right in here, and how this all feeds into our giant main pathway of metabolism. So proteins, amino acids, and then the catabolism. We're not going to talk about the biosynthesis of amino acids today. These chapters do talk about the metabolism of biomolecules and how the body synthesizes them. But we have really just been focusing on, in chapters 22, 24, and 25, the metabolism part. So the catabolism, not the anabolism part. So first off, amino acids. We introduced amino acids chapter 18. That was our chapter where we looked at protein structure. And so amino acids are essential for our body, mainly because they contain nitrogen. Okay, they have an amino group on the backbone, but some amino acids also have a nitrogen on their side chain. A few of them do, an amino group there. And that's beneficial because the body needs nitrogen for all different kinds of compounds. So the reason we break down proteins and amino acids is so that we can kind of extract that nitrogen, the amino group, off of amino acids and use them in the synthesis of other biological molecules. All right. So our body can make certain amino acids, but there are nine essential amino acids. So when we look at the pool of 20 amino acids, there are 9 that are considered essential. And that's at the very end of this, this lecture. The last slide has a table of those 9. And they're essential, again, because we have to obtain them from our diet. So the other 11 are considered non-essential because the body has a method and a means for synthesizing those. Okay, so the first thing I want you to know is that some amino acids are considered essential. We have to obtain those from our diet because we do not synthesize those. I want you to know which 9 are considered essential. Okay, if you know the 9 that are essential, then you know the others are non-essential. So for the exam, I do want you to know the 9 that are considered essential. And there's several bullet points of the deficiency of those amino acids and how they affect our body. That's the very last slide we'll look at today. So histamine is one of those amino acids, okay? Now, a deficiency in histamine, if you actually go to your textbook and look at kind of the cover page for chapter 25, it shows you a picture of somebody with rheumatoid arthritis. Anybody familiar with rheumatoid arthritis? What is arthritis?
Isn't it when your bones can't like bend and move the way they used to?
It's particularly with your joints. It's associated with a lot of joint pain. So people who have it really bad in their hands, they'll have a lot of like joint pain around like their knuckles and the bends of their fingers. And sometimes if it's so bad, your hand can completely like twist and become deformed essentially in ways that's very painful. So a deficiency of histamine is actually linked to or associated with rheumatoid arthritis, okay? And so sources of histamine, I kind of put right here on this picture. Meat, fish, legumes, nuts are all good sources. Salmon, I think I said fish already, but these are all good sources of histamine, okay? So I will want you to know the essential amino acids and then one thing that can happen if we're deficient in that amino acid. And that is on the last table of this lecture. All right, so before we actually get to the breakdown of amino acids and how they're used in the body, the first part of that metabolism is digestion. I mean, that's with any biological molecule that comes into our body. The first step in the process of metabolism is digestion. So digestion for proteins begins in the stomach by the protease pepsin. Oops, pepsin. pepsin. Now, this is actually formed as a zymogen, and it's called pepsinogen. Okay, so it's formed as a zymogen, pepsinogen, and then it's cleaved, and you get the active pepsin. And pepsin is active at a pH range of 1 to 2. We know we have HCl in our stomachs, and HCl is a very strong acid, so the pH of our stomachs is low. And pepsin is an abnormal protease because it's active at such a low pH. Normally, this kind of pH denatures any sort of enzyme, but pepsin is actually thrives here. So the proteins are broken into polypeptides by the hydrolysis of peptide bonds. And you guys should be really familiar with peptide bond hydrolysis now. But just as a reminder, here's a picture of it. Let's say we have some sort of polypeptide that extends this way and this way beyond the red squiggly lines. And then remember, R stands for the side chain. Okay, these are the side chains. So we don't know what amino acids these are, but in blue, with the kind of the red mark there, that's the peptide bond. It connects the carbon of the carbonyl group to the nitrogen, and that's the bond that breaks, and you get individual amino acids from that. Okay, it's a hydrolysis reaction. You use water, even though they don't have it written in, and the water breaks apart and hydrolyzes the peptide bond. Okay, so let me look at this figure really quick. So that kind of gets us to here, the mouth and pepsin, or sorry, the stomach, mouth, stomach, and pepsin. So then proteins are not entirely digested in the stomach. Then they move to the small intestine. Okay, so they're denatured and partially hydrolyzed by pepsin. Then they go to the small intestine where they're further worked on by other proteases. And the other proteases we're talking about are trypsin, chymotrypsin, and carboxypeptidase. So carboxypeptidase, just from reading the name of that, what do you think this enzyme specifically does to a polypeptide? If you had to guess.
Cleave it at the carboxyl group?
It cleaves off the carboxyl terminal amino acid. So for example, I'm just going to write the 1-letter codes. If we had like W and then L and then D and then G and then A. Okay, these are the 1-letter codes for amino acids. Obviously, the code on the one on the right, this is the carboxy. Oops. Or we call it the C terminal. The other side, remember, is the N terminal. So carboxypeptidase is going to cleave off the amino acid on the carboxy terminal side. Chymotrypsin and trypsin, do you guys remember where they cleave?
Doesn't the chymotrypsin cleave the aromatics?
On the carboxy terminal side of aromatics.
Well, all of them are on the carboxy terminal side. And then the trypsin cleaves at lysine arginine, right? Lysine.
Yes, positively charged amino acids, lysine, arginine, and sometimes histidine. Okay. So that's the small intestine worked on by proteases. So after that, we have individual amino acids. By the time they're done in the small intestine, now we've broken everything down into individual amino acids, a few dipeptides. Then they're transported across the lining of the small intestine into the bloodstream. Okay. And then they can be sent wherever they need to go. So that's digestion. Once we have individual amino acids, we can further break down the individual amino acids. That's called amino acid catabolism. And here's where we can start to pull off the nitrogen groups or pieces of the amino acid that we can use for the synthesis of other biological molecules. Okay. Now, let me say this. So all the time our body is regenerating. Do you guys understand? Like, what does that mean to you when I say our bodies are regenerating?
Like making new cells, like regenerating new cells and stuff.
So certain cells are dying off new cells are being made, certain biological molecules are being degraded so that new ones can be made, okay? So constantly regenerating. And this is good because sometimes things can break down on their own. Sometimes things can be made faulty. And so it's good that we're constantly recycling things. The body's really good at recycling. But with amino acids, there's sometimes when we break things down, we have to be careful too. Particularly with amino acids, we have to be careful because those amino groups on amino acids, that certain derivatives of those are toxic to the body. So ammonia and the ammonium ion are toxic to cells, okay? So we cannot store ammonium or ammonium ions in the body. We cannot store them, okay? So the body does not store nitrogen-containing compounds. So there's two fates for nitrogen-containing compounds or particular amino groups. They can either be converted to urea and excreted. Okay, so this happens, by the way, in the liver. And then for excretion, they're sent to the kidneys. Or we can take that amino group off and immediately use it to synthesize new nitrogen-containing compounds, okay? So the body constantly has, the way your book phrases it, and it's not a bad idea, this pool per se of amino acids. And those amino acids are constantly coming either from our diet and the digestion of proteins that we put into our body, or the degradation of old proteins as they break things down and recycle. So we constantly have this, and every cell, every tissue does this, but the synthesis of this new stuff and what we're going to get into, the catabolism, happens primarily in the liver, okay? And then again, it's either excreted through the kidneys, or we synthesize new biological molecules and put them back in the bloodstream and send them to other cells and tissues. All right, so I'm going to give you, this is the slide that I said I added. So you don't have this if you are using just your original slides, but this is just an overview slide. What we're about to get into, here's the steps of amino acid catabolism. The first thing we're going to do is take off the amino group. So there's a specific reaction, specific enzymes for that. Then we're going to use the removed amino group for the synthesis of new nitrogen compounds. So the next slide tells you several compounds that can be used for. Then we're going to talk about, okay, if we're not going to synthesize new compounds, we've got to excrete it. We have a particular cycle for that. So if we're not going to use it for the synthesis of new molecules, we're going to send it to the urea cycle. That's the title of it. And then also carbon. So we don't just want amino acids for their nitrogen. We also want them and recycle them and catabolize them or metabolize them for their carbon atoms. So we could also look at the fate of carbon from amino acid catabolism. And the major way we recycle these carbons is we send them into intermediates that fit into the citric acid cycle. Okay, so we recycle carbon to the citric acid cycle, we recycle nitrogen many ways, or we excrete it in the urea cycle. Okay, so that's the overview. Let's look at, before we get into the specific step number one, what reaction catalyzes the removal of the amino group. Here is the fate for nitrogen or carbon, kind of like I mentioned, but some specifics on this. So nitrogen can either be recycled into nitric oxide. Nitric oxide is actually a very interesting molecule chemically. It's actually a free radical. And what do we know about free radicals?
They can react with anything in the body.
It seems very damaging, right? We have antioxidants specifically for the removal of free radicals in our body. But nitric oxide is different. It's actually very helpful. It helps protect against like heart attacks. There's been several good uses for it. It's a messenger all throughout our body. So we need this molecule in our body. We don't get rid of nitric oxide. So that's one destination for the nitrogen from the amino groups. It can also be used to make hormones. Okay, those hormones could be different steroid type molecules. It could be insulin, glucagon type molecules, any sort of chemical messenger, neurotransmitters, coenzymes. So the nitrogen could be recycled into or synthesized into those molecules, heme groups, and then pyridine and pyrimidine for the synthesis of nucleic acids. Okay, so there's just kind of an array of molecules that we need the amino groups, the nitrogen from amino acids to build and make in the body. All right, carbon though can be incorporated in compounds like we said that enter the citric acid cycle, or they could be stored as triacylglycerols. Okay, so the process for that is we would take carbon, and this would be through lipogenesis. They could be converted into triacylglycerols. So we mentioned lipogenesis yesterday at the very end, but we did not get into the steps of lipogenesis. Okay, but that, I mean, think about how many carbon atoms are needed to synthesize fatty acids, right? Like I'm going to draw a picture of a fatty acid. It could be a really long chain. If the body is going to synthesize fatty acids, sometimes you got to have like 20 carbons. Okay. So lipogenesis is one route those carbon atoms can go to. It could be stored as glycogen. Okay, we know carbohydrates, glucose, fructose, mannose, all kinds of monosaccharides, disaccharides, those carbon atoms can be made for the biosynthesis of those and stored as glycogen if we need it, or they can be used to build new proteins as well. All right, so that's the destination for the carbon atoms. So speaking of amino acids and neurotransmitters, different molecules that amino acids can be turned into, I want to read this little question here. So this is actually going to ask you to pull from some of your enzyme knowledge. So serotonin is a monoamine neurotransmitter. And guess what? It looks a whole lot like tryptophan. Tryptophan is an amino acid that is a precursor to serotonin. So it's formed in the body from the amino acid tryptophan. What class of enzyme catalyzes each of the two steps that convert tryptophan to serotonin? So let's look right here. I'm going to zoom in on this. Tryptophan to serotonin, which is down here. Okay. So the first thing that is different, I'm going to erase this. The first thing that is different between tryptophan and then 5-hydroxytryptophan is that there is a hydroxy group on, I guess, carbon number 5 of that ring. So what sort of enzyme would catalyze this? What do you guys think? So the different classes and options are, you've got oxidoreductases, isomerases, hydrolases. We're not breaking anything apart though. We've got ligases and lyases. What does it look like to you? What's happening here? So this carbon right here, what is bonded to it? I see 3 bonds, so it must have one hidden hydrogen that we don't see. And then that gets replaced with a carbon oxygen bond, the OH. So how would you classify that? We're looking at this bond right here. That's the new bond.
It's not hydrolase, is it?
It's not hydrolase. We're not hydrolyzing anything or breaking anything apart.
Is it octase? Or
Oxidase. Oxidase. Yeah, this is an oxidation reaction. So this would be an oxidoreductase. And the subclass there specifically would be an oxidase. Okay. So we have this new carbon-oxygen bond, so we oxidize that carbon. Okay, now look at the difference between 5-hydroxytryptophan and serotonin. And we take off a CO2. Now, we are not using a coenzyme here, so it's not a dehydrogenase. We're taking off a CO2 from the backbone of tryptophan to leave us with serotonin. What kind of enzyme would do this? Here's our enzyme table. So we look through these. Carboxylase catalyzes, no, it adds CO2. It doesn't take off CO2. So it's not a ligase. Liases, you've got decarboxylase catalyze the removal of CO2. Could it be that?
Yes.
For sure. That's what we're doing. We're taking off CO2. So this is a decarboxylase. Okay. So on the exam, I could give you a reaction like this and say, you know, here's one way an amino acid is used to synthesize this biological molecule. And by analyzing what is added to or taken away from the substrate, you should at least be able to classify it into a main group. Okay. If you're not sure what subgroup it is, you should at least be able to classify it into a main group. So a decarboxylase is a ligase. Sorry. I get those two mixed up all the time. Okay. All right, so that's step one. That's just showing you how the nitrogens, we haven't specifically looked at carbons yet, but how an example of how nitrogens can be used in the synthesis of different biological molecules. So here is where we're going to get into the specifics of amino acid catabolism. So there's a series of reactions that take place if we're getting ready to excrete or get rid of the amino groups from amino acids, or if we're getting ready to pull them off and use them in the synthesis of other things. So the first step is called a transamination. Kind of fun to say. Transamination, the amino group and a keto group from an alpha keto acid swap. This is conducted by transaminase enzymes. So it's kind of nice because the name of the enzyme matches the name of the reaction. And guess what? When you saw alpha keto, I bet you thought of a particular molecule. It is we've heard of alpha keto before, alpha keto glutarate. This is an intermediate in the citric acid cycle. If you're like, I know I've heard this word before, but I don't know where I heard it from. Anybody ever run into somebody before in like a grocery store and you're like, I know that I know you, I just don't know where I know you from? That literally happened to me the other day. Had the weirdest conversation with somebody. Anybody ever meet people? Me and my husband have a word for people that kind of just seem like a little, there's nothing wrong with them, but it's just hard to have, we call them space cadets. It was a total space cadet conversation of somebody that I had like met years ago. It was very, okay, anyways, back to alpha keto glutarate. So you should recognize this from the citric acid cycle. And this is the common amino group receptor. So most transaminase, it's not the only one, but it's like the majority of the time that a transaminase happens, alpha keto glutarate is going to be the alpha keto acid that is accepting the amino group. So here's an example. Let's say we have this amino acid and here's the side chain. We don't know what side chain, this could be several different amino acids. Okay, all of them undergo transaminases via their own little pathways, but the process, the overall principles guiding the process are the same. Okay, so it could be any amino acid, but we have some sort of alpha keto acid as well. These are our two reactants, and what makes it the alpha keto is this carbonyl group right here, and we have this carboxylate group right next to it. Okay, so there's the alpha keto acid. If you look just down below, here's alpha keto glutarate. So again, you see the carbonyl group and the carboxylate group right next to it. It's a ketone. Why? Because of this right here. The carbonyl carbon is bonded to a carbon on both sides. Remember, that's what makes it a ketone. All right, so the alpha keto glutarate, and the reason it's called an alpha, do you guys remember where that term alpha comes from? If we were to draw this out entirely, what it would look like is this. Right, and then the R. So this carbon right here is the alpha carbon to the carboxyl group or carbonate group. So that's why it's called an alpha. And then the word keto comes from the fact that this is a ketone functional group. All right, so what a transaminase does is it simply switches the red and the orange groups, or it might look yellow to you. It switches the red and the yellow or the orange groups. So the double bond to oxygen is going to switch over to the this carbon right here. So this group is going to jump over to here. And then the NH3 plus group is going to jump over to that. They're just going to switch places. We're going to transfer those two groups, transfer an amino group. So that's what you get when you finish. And this alpha keto, now we have another alpha keto acid. We call it number 2 though, it's on the product side. And then we have a different amino acid. So it feels a little bit redundant because your products are very similar to your reactants, but there's purpose to this. So I want to show you the main example. Since we said alpha ketoglutarate is usually this amine group receptor, the one accepting this new amine group, I want to look at a specific example with alpha ketoglutarate as the receptor. So if we have alanine, and we know alanine simply has a methyl group as its side chain. So there's the side chain. So this is the alpha carbon in alanine, and we're going to switch the NH3 and the double bond to oxygen. So when we get done, if this is alpha ketoglutarate, when we get done, we have turned alpha ketoglutarate into glutamate. If you look at this closely, this is the alpha carbon of glutamate. So this whole part would be like the backbone. And then this is the side chain, a CH2, CH2, and a COO minus. This would be like the R group of glutamate. So when alpha ketoglutarate is the dominant amino group receptor, we make a whole lot of glutamate. That's just the byproduct of this reaction, okay? And then pyruvate as well, specifically if you have alanine, but if your amino acid's not alanine, you won't end up with pyruvate. This was just the product because that's what's derived from alanine, okay? So that is step 1 transamination. So you definitely need to walk through a transamination for the exam, okay? Know which groups switch. If I gave you a different amino acid besides alanine, and if I gave you alpha ketoglutarate, you should be able to write the products. You would for sure get glutamate, and then whatever is left over from your amino acid when you have that carbon, alpha carbon now double bonded to an oxygen. And if you notice, so I want to zoom in on the enzyme. It's called an aminotransferase, but we just put the name of whichever amino acid is involved in front. So because each of them have their own individual pathways, each of them have their own individual aminotransferase. So you could have glutamine aminotransferase, you could have proline aminotransferase, you could have whatever the amino acid is, you say the name of the amino acid and then you say the aminotransferase. Okay, the next step after transamination is called oxidative deamination. So what we're going to do now is the amino group is going to be removed as an ammonium ion off of glutamate. And again, we're just using glutamate here because alpha ketoglutarate is usually the amino acid receptor. And when it accepts an amino group, we get glutamate. So we end up with a whole lot of glutamate in the body. And we're going to use water. And we're going to use a coenzyme, NAD plus or NADP plus. And this is a dehydrogenase reaction. So we have a dehydrogenase enzyme. And we're going to end up with, so this is just going to be cleaved off right here. Okay. And that carbon is now, so this carbon right here, kind of the alpha carbon, this is still the alpha carbon, is now going to be double bonded to oxygen. All right, and then in the process, one of the byproducts is NH4+. Now remember, this is a toxic substance to the body. So we can't just let it hang out by itself. We're immediately going to send it to the urea cycle. But we have to get that ammonium group off so that we can, this destination is directly to the urea cycle. And this cycle is what we're about to get into in just a little bit. Okay, so 2 steps. These are kind of like the precursor or the preparation steps for the urea cycle. Transamination. Now, just because you do transamination doesn't mean you're going to send things to the urea cycle. You could still synthesize other amino acids or biological molecules at that point. But once you do oxidative deamination, you're committed to the urea cycle. Okay, that ammonium ion is going to go right to the urea cycle. Okay, so after transamination, you can still use the nitrogen group to make other biomolecules. Does not have to go to the urea cycle, but if oxidative deamination happens right after that, the destination is the urea cycle. Okay, questions so far? Could you walk through on your homework? You'll have more practice for this, a transamination reaction and an oxidative deamination. Okay, we're calling it deamination because we're pulling the amine group, the nitrogen containing group, off. So let's actually practice that really quick. What is the structure of the alpha keto acid formed from the transamination of the amino acid phenylalanine? Phenylalanine. Do you guys remember how to draw phenylalanine? I bet somebody can tell me. It's an aromatic. I'm going to draw, I know we have an alpha carbon with a hydrogen, and what do I draw next?
I might be wrong, but don't you just do an aromatic? This one doesn't have like the alcohol group attached, right?
No, that's tyrosine. Yep,
Okay.
This one's simple. So like this. If you are ever confused, you know, you can always, it's not like reading my, maybe I have to be, okay. You can always jump down to your, well, you can always jump over to your amino acid table. You should always have these things handy. Remember? There's one thing though we're missing on the side chain, the CH2 group. So it is an aromatic. There's no functional group on the other side, but we do have this CH2 group connecting our backbone to the aromatic group. So we need to add that. Okay, now we got phenylalanine. Okay, so we're gonna have some sort of alpha keto acid. Let's just draw alpha keto glutarate. Okay, alpha keto glutarate. So we're just gonna draw this molecule right here. Okay. So now what happens? Let's highlight the parts that switch, this NH3 group and this carbon oxygen double bond. The carbon doesn't go anywhere, but I guess I should highlight like just this part, just the double bond to oxygen. So those are going to switch. So now you should be able to draw the product. So just redraw phenylalanine, but on the alpha carbon here, now we have a double bond to oxygen. We still have our side chain. And we still have our carboxyl group. And then we get glutamate. I'm gonna have to draw small. Okay, so these would be the products if we did a transamination reaction on phenylalanine. So could we name the enzyme that does this? Remember, we just said we use the name of the amino acid, so phenylalanine. Amino transferase. It's always going to be an amino transferase. We just put the name of the amino acid that it did the reaction on in front. Phenylalanine amino transferase would be the enzyme.
On the exam, do you want us to name the reaction?
Like as a transamination.
Or like title it like the phenylalanine?
Oh, name the enzyme.
Mhm.
Yeah, because you know it's going to be an amino transferase, and then just depending on what amino acid you're given, you would just say it's that amino transferase.
Okay, thanks.
Yep. Okay, so now let's go back to oxidative deamination. At the end of that, remember we have this ammonium ion, NH4+. And this is going to go to the urea cycle. So what is the urea cycle? How is it used? Why is it important? This is how our body excretes nitrogen, okay, if we don't need it. So we've mentioned this, ammonium as well as ammonia are toxic to mammals, and we cannot fish. Our fish are lucky. They can just directly diffuse ammonium ions and ammonia out through their gills, okay? We don't have that luxury, okay? Not just because we don't have gills, but because if we were to directly excrete ammonia, we would dehydrate ourselves, okay? It would just take so much water to dilute ammonia. Maybe we just have so much of it coming through the urea cycle. So the best way for our body to do this is to convert it to a different compound. So we convert it to urea. Oops, urea. And then we can excrete urea without dehydrating ourselves. So urea, by the way, I'm going to draw it. It's right down on the next slide, but it's a carbon double bonded to an oxygen. And then we have NH2 and NH2. Okay, so notice that there's no charge on urea, unlike the ammonium ion. And that's the reason we can safely excrete it without dehydrating ourselves. When you have your blood or your urine is full of a lot of salts, things that are positively charged, sodium, chloride, ammonium, it takes a lot of water to dilute these things and keep them at a regular concentration or a manageable concentration. And getting rid of water in the body is what obviously can lead to dehydration. So the fact that this has no charge on it helps us excrete it without dehydrating ourselves. All right, so before we just jump into the urea cycle, by the way, I actually do want to zoom in on this figure for a second. So this is what we've walked through so far, transamination and oxidative deamination. The parts that are highlighted in pink, that's tracking the amine group, the nitrogen in the amine group, and how it's being passed from each intermediate. So obviously the amino acid starts off with the nitrogen, and then the nitrogen via an amine group, an NH3+, is passed off to ultimately glutamate. Now, glutamate is made from alpha ketoglutarate, so that is what is making glutarate, but the nitrogen is passed to glutamate in that transamination reaction. Okay, the amino acid is turned into an alpha keto acid. So the way to read this is that here would be reactant 1, here would be the corresponding product, here would be reactant 2, here would be the corresponding product 2. So these are the reactions. It's just that the nitrogen is passed from the alpha amino acid to glutamate, and then in oxidative deamination, it's passed to NADPH is used to generate a free amino group. It's coupled to the reaction. And we end up with a free ammonium ion. Free ammonium ion. Okay. Once we have the free ammonium ion, it's going to feed right into the urea cycle, but we're going to have to react it. Here it shows CO2, but this CO2 actually makes bicarb HCO3-. This CO2 comes from the citric acid cycle. So you know how in steps 3 and 4 of the citric acid cycle, we make CO2? That CO2 comes here, it reacts and makes bicarb, and that reacts with NH4 plus in this reaction that you see right here. It's a precursory step, formation of what's called carbamoyl phosphate. And that is the starting material for the urea cycle. Okay, so we take this, what's happening here? It's actually quite simple. Maybe it looks complex, but we have the ammonium ion and we have bicarb. Why is bicarb important? I've really not talked about bicarb in this class so far, but we did talk about it a whole lot in 1402. Why is bicarb important? So this is a polyatomic anion that is part of our blood buffer system. The way we get bicarb from CO2 is like this. CO2 from the citric acid cycle from our cells makes CO2 just as waste, plus water makes H2CO3, carbonic acid, and that immediately breaks down into bicarb and H plus. Okay, carbonic acid is very unstable, so that's where we get bicarb from CO2. This is our blood buffer system, by the way. Okay, so that's where bicarb comes from. It's just in the figure, it shows you CO2. So we have to use 2 ATP. Wow, that's expensive, right? Energetically. 2 ATP to power this reaction. And essentially we're taking a phosphate group, we're taking bicarb, and we're taking the ammonium ion, and we're mushing them all together. So the ammonium ion is right here, that's part of the carbamoyl phosphate. The bicarb right here now becomes this CO2 part. So we lose the hydrogen and we lose an oxygen. Those are recycled into water over here. And then we have the phosphate group from ATP is added on. So we just mush 3 parts together to get this substrate that goes into the urea cycle. And we get 1 phosphate group left over. But the main thing I want you to know is how these things come together to make carbamoyl phosphate. And then we're going to see how it goes into the recycle. Any questions so far? Any questions on how to read this figure and like the how to track where the nitrogen group is going?
So I know what you said, like it makes sense, but can you like I don't know how to say this. Can you like take off the colors and show me one more time how like
Yeah, let's walk through this again.
Okay, thank you.
Yeah. The bigger eraser. Okay. So the nitrogen, if this is amino acid, we know it has some sort of NH3 at physiological pH, it'll be NH3. Maybe at a different pH, it would be NH2. But at physiological pH, we've got some sort of NH3 group attached to this amino acid. When transamination happens, this NH3 group is transferred to ultimately what we make is glutamate. Now, glutamate, the 2 reactants, so this would be like reactant 1 and this would be like reactant 2. And they come together and we just transfer the amine group from that amino acid to the product from the alpha ketoglutarate or the alpha keto acid. So this would be product 1, the corresponding product, this would be product 2. So we alpha ketoglutarate ultimately is making glutamate, but we've transferred the amine group from here to this guy, and in the process, that's how we produce glutamate. So that's why on the molecule, what I'm calling R1, the amino acid, that's why that's highlighted pink, because it contains the nitrogen group. But when the transamination is done, now the glutamate is the one that has the nitrogen group. Okay, then oxidative deamination is going to couple the oxidation of glutamate essentially with the reduction of NAD plus or NADP plus. So we're going to reduce the coenzyme and we're going to end up with a free ammonium ion. So now the NH3 has been pulled off of glutamate and we remake alpha ketoglutarate in that process. That's why this arrow is curved upwards. We pull off the NH3 group and we just have it right here. And what's left over from glutamate when we pull out that NH3 group is its original substrate, alpha ketoglutarate. So this is kind of a circular thing here. It just kind of causes the cycle to happen again and again and again. We just remake alpha ketoglutarate when we couple it to the reduction of this coenzyme. And then we end up with the free ammonium ion, and that feeds right into the urea cycle. Hope that helps. The whole curved arrow notation is just the way we show coupled reactions happening. And it just so happens these are cycles because you remake your original substrate and it can just keep happening again and again. Any other questions on this figure? Please don't ever hesitate to ask me to re-walk through something. Okay, so the urea cycle is right here. So we're going to start up at the top. There's 4 steps. We've got the carbamoyl addition is the first step. So just like the citric acid cycle, this is a circular or a closed pathway. Okay, I don't want you don't need to know the enzymes for all of this, but I do want you to track where the nitrogens come from and where they go. That's the whole point of this, is we're seeing what happens to nitrogen. So the group I want you to focus on right up at the top here is right here. Okay, this is the carbamoyl phosphate is the group we just made in the slide up above. And so now we made it from NH4 plus and CO2, and it's oriented differently. I know they're on the left up here and put them on the right down here. I'm sorry. I hope that doesn't confuse you, but here's what we have, carbamoyl phosphate. And it is going to react with something called, this is a hard word to say actually, ornithine. Ornithine. And it's an amino acid. It's one we've never heard of because it's not used to make proteins. Okay, so there are other amino acids besides the 20 we learned, but it's a non-protein amino acid. It's kind of weird to say. It's a non-protein amino acid. So it looks a whole lot like lysine, but it's just one CH2 group shorter on the side chain. Okay, so what's going to happen is this part boxed in red, the carbonyl group with the amine on it, is going to be transferred to this nitrogen right here. So this carbon and this nitrogen are going to form a bond together, which is what you see right here. And we're going to lose this phosphate group in the process. Okay, that's just going to be cleaved off. So we're just going to cleave the carbamoyl phosphate group off, and we're going to bind the carbon to this ornithine amino acid. And now we have something called citrulline, which is also a non-protein amino acid. So I'm going to highlight the bond that formed. I guess I'll highlight it in pink since that's the color I'm on. Sorry, it looks like I highlighted 2 bonds. Okay, that's the bond that formed. And again, the atoms highlighted in red or colored red, these are from initially the carbamoyl phosphate. So in this step, we have now added the first nitrogen that's headed for excretion to the urea cycle. Okay, so, and that is this nitrogen right there. That one's going to be excreted from the body. It's going to be incorporated into urea. Because there is another nitrogen, right? Down here and down here. That one stays where it is. It's the nitrogen that was added on from the carbamoyl phosphate. That's the nitrogen that's going to be excreted from the body. Okay, then so that's step 1, citrulline or citrulline. Once we have step 1 done, step 2 is we're going to have aspartate. We know that is a protein amino acid. Aspartate, or people say aspartate. This is literally just going to be, we're just going to add it on to citrulline or citrulline. Okay, we're just going to add this whole thing on. So what happens is, oh, what did I do? Okay, here I am. This oxygen, I'm gonna color it in blue. This oxygen right here, this is going to go away. We're going to rip that off, and we're going to bind aspartate to the carbon via the nitrogen group. Okay, so instead of a carbon oxygen bond, I'm going to highlight this in pink now. This is the new bond that's formed right here. So the NH2 group, the one with the star by it, that one's still there, the one in red. That one didn't go anywhere. We just pulled the oxygen off and we put aspartate on. Okay. So now we have added the second nitrogen that's going to be excreted in as urea to the urea cycle. And that one, I'll just put a star, is this nitrogen right here. So now we have two nitrogens bonded to the same carbon. These nitrogens plus the carbon, that's going to be excreted from the body. So this is how the body gets rid of excess nitrogen.
So are all the nitrogens with the stars by them are the ones that are excreted?
Those are the two that are excreted.
Well, there's three. You have stars by.
They're the same nitrogens. So this nitrogen is just this one right here that still has the star by it. It's the one in red.
Yeah.
And then this nitrogen in blue is the same as this nitrogen right here.
In blue. Okay. Yeah.
So
yeah.
what I could do is I could put a 1 and a 2, I guess. That might help. Will that help you, Robin? If I say this is nitrogen number 1,
Mhm.
this is nitrogen number 2.
Yeah. Thank you.
2. So in the process of doing this requires ATP, by the way. We're going to hydrolyze ATP all the way to AMP. So we take off 2 phosphate groups. So this requires a lot of energy. But again, you've seen this. It requires 2 ATP up here and another ATP down here. This has required like 3 ATP so far. And again, this is what you see, and we're not generating energy from this cycle, by the way, so it's a very costly cycle. We don't get any ATP back out of it. But the same reason I mentioned yesterday, this is a common theme in metabolism, is that to metabolize something, to break it down, we have to destabilize the substrate. So these first 2 steps, we're adding on, we're just essentially adding things on to ornithine to destabilize it. Because here's what we're about to do. We're about to cleave some stuff off. So these are called destabilization steps. So now look how big this arginosuccinate is. It's huge, right? And it's very unstable. So what we're going to do is we're going to cleave off. The bond we cleave is right here. We're going to cleave this bond right there. And that actually is fumarate. Anybody remember what fumarate is? And the citric acid cycle. I'm going to abbreviate citric acid cycle. So that can just go right back to the citric acid cycle. And then we have that's the end. That's actually step 3. After we cleave this off, what we have left is just arginine. We know what arginine is, so we just made arginine. But we're not going to use arginine. We're going to further cleave something off. We're going to now cleave urea off. So again, pay attention to the parts in the red and the blue. That's the part that comes off. Okay, now we use water to do this. So technically it's a hydrolysis reaction. We're not cleaving a peptide bond here, but we are breaking off a piece from the R group. So this is the side chain. And we just cleave off everything in red and blue. So this is nitrogen number 1, this is nitrogen number 2, and that carbon goes with it. And because we're adding water, that's where this double bond of the oxygen comes from water. And that's urea. Now we've made urea. And that just it goes to the kidneys and it's excreted from there. So this is really important because, like we said, this is part of our body's defense system from molecules that are toxic, like ammonia and ammonium. We have to have a way to get rid of them. So we break them down. Ultimately, we synthesize urea and then the body excretes that. So this is one fate of amino acid catabolism. For the nitrogens. So the steps, we have the carbamoyl addition synthesis, the cleavage step, that's what I drew here. This is the cleavage. And then the hydrolysis step is step 4 because we're adding water. Okay, so I want you to know these 4 steps. This is not a ginormous pathway if you consider like glycolysis or the citric acid cycle where there's 8 or 10 steps. There's just 4 steps here. So I want you to know these 4 steps. If I gave you the intermediates, I would want you to be able to name them and ultimately identify the nitrogens that end up in urea. Okay. If I gave you an open-ended question, I said, tell me where the nitrogens come from and how the body excretes them or whatever. I would want you to be able to, in summary form, tell me, well, this is the gist of the urea cycle. We make the carbamoyl phosphate. We add that on to ornithine. We add aspartate. There's a cleavage step and a hydrolysis, and we end up with urea. Okay, that would be like a summary. And then that's sent to the kidneys where it is excreted. Okay, questions on the urea cycle? Okay, how much time do we have left? I think we only have 3 slides. Yeah, we got time. So this is a practice problem. Fumarate from step 3 of the urea cycle can be recycled to aspartate to be reused in step 2. So we can literally take this guy and recycle him right back to here so we can just continue the cycle. Oh, by the way, after we cleave off urea, we remake our initial reactant, ornithine, and the cycle just is ready to go again. Okay. So it's a 3-step process. And I want you guys to tell me, I want y'all to look at this. Let me see if I can launch. I don't think I can just, I don't have one made. I would have loved to have written a poll question for this, but I want you guys to look at these and tell me your options are, I want you to classify these reactions. Classify each reaction as either an oxidation, a reduction, transamination, elimination, or an addition. So I want you to look at fumarate and look at we add water and we make malate. So the thing that's different is, I guess I could highlight it. Right here, we have this OH group bonded to that carbon. We broke the double bond in the process. What kind of reaction would you say this is?
Is it oxidation?
I can't fault you for that, Madison. We are oxidizing this carbon. So honestly, I would accept 2 classifications here. I would not tell you you're wrong if you said this is oxidation, but it's also what?
Addition.
It is an addition reaction. So when we first introduced alkenes, we talked about all different kinds of addition reactions. Hydration, this is a hydration addition reaction, but you're not wrong, Madison. We're also oxidizing this carbon. I recognize that. Everybody see how it's oxidation? Everybody see how it's addition? The water, from the water, the OH goes right there, and then the hydrogen goes right down that carbon from water. Okay, what about the next step? So now we have malate, and then we convert it to oxaloacetate. So look at what's different. Let's highlight. We have this OH group, and now that's converted to a carbonyl group. And we're using a coenzyme. So we saw this happen like 3 different times in the citric acid cycle. So we're taking off that hydrogen there and that hydrogen there, and we're making a double bond to the oxygen.
Is it reduction?
So reduction increases the number of carbon hydrogen bonds, or it's the gain of electrons, or it decreases carbon oxygen bonds. So we're actually increasing carbon oxygen bonds here. We go from this having 1 carbon oxygen bond to 2 carbon oxygen bonds. So this would for sure be oxidation. Okay, what about the last one? What is different? I'm gonna try to highlight it in pink. That carbonyl carbon now has an amino group added to it. You should all for sure get this one.
Is it transamination?
Transamination. This is a transamination reaction. Okay, so we have the glutamate reacts with it. You get recycle alpha ketoglutarate and you end up with aspartate. So that's how fumarate is turned into aspartate. And then it can just feed back into step 2 of the urea cycle. Okay. I think this is good practice. I tend to always put reaction question like this on the exam, and they've always been worth like 12 points. Looking at the reactant and the product, either classifying what kind of reaction or what kind of enzyme would catalyze this. So this would be catalyzed by an amino transferase, specifically glutamate, because that's the starting material, or at least the amino acid here. This is an alpha keto acid. This would be a dehydrogenase. This would be an oxido. Sorry, my handwriting is very bad today. Maybe that's every day. Actually, I'm going back on my statement there. Here's why. No, I'm not. I think we're good. No, actually I am. This is for sure a hydratase. See how you take the double bond, you add water, and it's exactly what we got in that reaction. We got the OH on one carbon and the hydrogen on the other. Yeah, this is a hydratase. Yes, oxidation is happening, but the type of enzyme that would, because we're adding water, this would for sure be a hydratase. Okay, any questions on that?
You said you usually put something like this on the test every year?
I mean, I have the past 3, right? I actually don't quite know if something like this would be on the exam, but this is really, really great practice, either for exam 4 or on the final. You'll probably see something similar to this. I may not give you 3 in a row where it's all like one similar process, but I think this is a great practice problem. It just brings a lot of our knowledge of different things, functional groups, enzymes, pathways, all together in one question, which is why they're hard. Okay, 2 slides left and then we're done. So we spent this whole lecture talking about the nitrogen atom. But remember we said that the carbon atom, we don't just want amino acids for their nitrogen, we want them for their carbons too, and those can be recycled. The main place they go is they feed right into intermediates in the citric acid cycle. Okay, so here is the citric acid cycle. We have pyruvate, acetyl-CoA. We know acetyl-CoA reacts with oxaloacetate, and then you begin the cycle. It's a closed cycle. There's some other things I'm going to talk about, but in the blue and the pink boxes, you see all the different amino acids. I think that's all of them, actually. I have not counted. You see some of them twice. Okay, so for example, you see like phenylalanine. Well, I should probably highlight it. I'll just put a star by it. You see phenyl. Oh, I'm on highlight. You see phenylalanine right here, and then you see it over here. So that's okay. You'll see some of them can be both blue and pink, and I'm going to explain what blue and pink means in a minute. But essentially, all amino acids can feed into the citric acid cycle or a pathway that the citric acid cycle comes from or feeds into. So let's talk about the pink amino acids in the pink boxes. So isoleucine, leucine, threonine, and lysine, and then also down here you have leucine, lysine, phenylalanine, tyrosine, tryptophan. These can all be synthesized into via pathways that we're not going to talk through, but they can all be converted to either acetyl-CoA or acetoacyl-CoA, which is just a derivative of acetyl-CoA. Or sorry, acetyl-CoA. It's a great word to say. And these can feed into the citric acid cycle, or this acetyl-CoA can be turned into ketone bodies. Follow that route. So because of that, because these can feed into ketogenesis, essentially is kind of the formal name of that path that makes the ketone bodies, and we talked about ketone bodies and what they can do to our blood the other day. It can lead to ketoacidosis, which could be fatal because it drops your blood pH. All of these amino acids are known as ketogenic amino acids. There's a homework question on this, by the way. Ketogenic amino acids. If you notice, all the amino acids in blue, via all different pathways that we're not going to get into, can make almost any intermediate in the citric acid cycle or pyruvate itself, which feeds into the citric acid cycle. Okay. Now, all of these intermediates follow the cycle around clockwise to make ultimately oxaloacetate. Oxaloacetate can feed into gluconeogenesis. So I'm going to jump over to chapter 22 really quick, because at the very, very bottom, we talked about gluconeogenesis right here. And oxaloacetate is, boom, number 2, the second step in gluconeogenesis, which ultimately goes back to make glucose. Okay, gluconeogenesis is the creation of glucose. So that can feed into gluconeogenesis, make glucose. So we call these amino acids glucogenic amino acids. So on your homework, I think it asks you to give some examples of either a ketogenic amino acid or which amino acids are ketogenic, which are glucogenic. This figure right here is how you know. And this is another prime example of how the body links all these different pathways together so that 1, we don't waste resources, and 2, we can quickly adjust to different conditions. Let's say we need more glucose. There's so many different ways the body can make that. Let's say we have too much acetyl-CoA. We have methods and means for handling it as safely as possible. Okay, any questions there? And again, yes, some can be both. Phenylalanine is an example. Tryptophan can be both glucogenic and ketogenic. Isoleucine, several of them can be both. I'm trying to think if I see one. I think lysine can only be ketogenic is what it looks like. So there's some that are only one or the other, but several of them are both. Okay, last slide here. So there's a whole portion of this chapter we're just not getting into, which is the biosynthesis of amino acids. So we have a way to make amino acids, and therefore they're not considered essential to a body because we don't have to get them from our diet. We can make them in-house. So we're not going to go into the biosynthesis of non-essential amino acids, but you do need to know which amino acids are essential and non-essential. And the easiest way to remember that is to know the 9 that are essential. And we have to obtain them from our diet. So this is what I was mentioning in the very first slide, histamine. Deficiency in histamine has a possible link to rheumatoid arthritis. That's what RA stands for, rheumatoid arthritis. There's other deficiencies you can see there. You can see why they're necessary, what they do in the body. So here's what I'll tell you. I think I said you do need to know all 9 of these, but as far as like the roles and the deficiencies, I want you to pick 3 amino acids, 3 essential amino acids, and know their role and their deficiency, like what happens if you're deficient in that amino acid. So you don't need to, I'm not going to ask you to memorize the role and deficiency causes that can happen or effects for all 9. You need to know the names of the 9, but you only need to, I only want you to be able to give me examples for deficiencies of 3. You can pick which 3.
Does this table have the amino acids, the essential amino acids?
These are the essential amino acids.
Okay. I only have 7 on my table. Should I have 9?
Yeah, me too.
Well, you have 3 right here.
Sorry. One of them has 3 and one.
Yeah.
Oh, okay. I see.
So isoleucine, leucine, and valine are all very similar, non-polar, neutral. So their effects are very similar. Now they have some differences down here, like leucine and valine specifically, but yeah, if you count both leucine and valine as well, you get 9. Okay. So that kind of puts us at time for the day. And that's 2450. You guys should feel very proud of